Okay, so this is a question for Rocky Foch, who asked for a little bit of help regarding some numeracy and data handling in higher human biology, which I assume is going to be undertaken this year. So it starts soon. There's many different types, and I'm just going to use this question here to give an example of the types of question you can expect in either higher biology or higher human biology. If you're wanting to have a look at this question, you'll find it in the past papers. It's this year's paper, so the one sat in May, and it's paper 1, the multiple choice, and it's question number 23. Now, I don't think the marking schemes have been released just yet for it, but I'll take you through this question and I'll break it down to you so it's relatively simple. So, key things that I'm going to take here is I'm going to give you all the information and break it down so you don't make the common errors that people sort of fall into the traps of going for, especially when you look at these double graphs. So the first thing I'm going to do is I'm going to take a highlighter and I'm just going to simply highlight the context of the question. Okay, so the context is this graph here shows concentrations of T lymphocytes and HIV in the blood of an individual following infection with HIV. Okay, so nothing that's really going to help you answer the question here, but it sort of frames the question and gives you a bit of context to it. Now, the next thing we're going to do is we're going to ignore the graph, and using a different coloured highlighter, we're going to go and highlight the actual question. Okay, so the question is, which row in the table shows the concentration of T lymphocytes and HIV in the blood of the individual 3 years since they were infected with HIV? Okay, so from that there, from the question, we're going to read our answer from the graph from this point here, right? So hopefully you're following that so far. Nothing to be scared of, just simply reading the question and thinking about the context of it. Now, what we're going to do next is we're going to go to the key, and I'm going to take another highlighter for the key, and for the left hand side of the graph I'm going to highlight T lymphocyte concentration, which is here, concentration of T lymphocytes in blood number per millimeters cubed. So the reason I'm going to keep this in blue is to stop you confusing yourself. So whenever you are dealing with T lymphocyte concentration, you're going to use the blue highlighter, right? Which also means we're going to use the blue axis, okay, which I've now coloured in for you. As well as that, you'll notice also that the dotted line is goes hand in hand with T lymphocyte concentration, so I'm going to highlight that as well. Whenever you are doing any reading, okay, of T lymphocyte concentration, because I've highlighted the dashed line, I'm always going to be reading it from the left hand side. Now conversely, I'm going to take a different highlighter. I'm going to select red, and what I'm going to do is highlight HIV concentration in the similar manner. So highlight this here, it's the information. Then I'm going to highlight the right hand side axis, and then I'm going to highlight the solid black line, which is going to give you that information. Right, so at this point here, make sure that whenever you are reading from the HIV concentration, you're only dealing with the red. The blue parts simply do not exist. And vice versa, if you're reading for T lymphocyte concentration, you're only dealing with the blue. So for example, a common mistake if you don't use this colouring technique is you might use the black line, okay, and read it from the blue scale, right? That's a complete no-no, okay, because that's where people get marked wrong and that's where they get confused. Right, so going back to the question, Which row in the table shows the concentration of T lymphocytes and HIV in the blood of the individual 3 years since they were infected with HIV? So on the left hand side we've got concentration of T lymphocytes in the blood, and then the right hand side we've got concentration of HIV in the blood. For this I'm going to take a solid black line, sorry, not a solid black line, a solid line, and the first one I'm going to do is it's going to be the concentration of T lymphocytes. Okay, so I'm only going to be using the blue part of the graph, not the red. Just ignore it just now. We're only going to focus on the blue. So as it's involving year 3 since infected with HIV, that is where we're going to get our marker. So I'm going to take this line and I'm going to go from 3 years up to the point of the graph that meets that dashed line. Remember, we're only dealing with blue, so you do not go to the red scale here, you go back to the blue scale. And you can see that it hits perfectly on 100. So with this in mind, what you can already do here is you can take away by the process of elimination 2 of the answers as being incorrect. So straight away what we can do here is we can take out B and then we can take out D, right? So that leaves us with working out the concentration of HIV in blood, and the 2 options left is 50 or 500, right? So to get this final part of this question, what I'm going to do is I'm going to take a solid red. Again, I'm going to go up from 3 because it's 3 years since they were infected with HIV. Go follow up from the 3 to where we meet the red. Okay, and then we don't go to the left, we don't go to the blue side, we're only dealing with the red part of the graph. So go into the red scale, we'll go over here, and you'll see that it hits perfectly on 50. Okay, so with that in mind, the answer to this question is going to be A. If you don't use this colour technique, it's so easy to sort of fall into a sort of hypnotic trance and go from here to the left, and that would give you 500, guiding you towards C. But C ultimately is wrong because we don't deal with the red part of the graph and the blue. We don't mix them up at all, and that is how we make sure we keep ourselves on the right track. So use those highlighters, use coloured pencils. It's an easy and effective way of learning how to read these graphs. Now this explanations maybe took quite some time, but the more you practice doing these, the easier they get, and who knows, you might not even need to use highlighters, coloured pencils or whatever. You might be able to just do it off the top of your head, just reading the graph without doing any of the highlighting or anything like that. Okay, so I hope this helps, Rocky Fosh, and if there's any other questions that you can think I can help you with, then please do send me a message if I can be of any use. Okay, thank you.